I take one stem cell out of a human, and a stem cell is an embryonic cell. So do you have stem cells? Of course you have stem cells. Why? If you're here listening to us and watching Chris and I on this program, then you have stem cells, because if you didn't have stem cells, you'd be dead very shortly in your youth. I go, why? A human body is not a single entity. It's made out of 50 trillion cells. And every day, hundreds of billions, these are numbers that are astronomical, hundreds of billions of cells are dying every day in your body. But you're still here. And I go, yeah, because mixed in your population of cells are stem cells. And stem cells are the embryonic cells in your body that will replace all the cells that are dying. Stem cells are very important because they're associated with cancer when they get out of control, okay? So what am I doing? I got a stem cell, one stem cell, and I put it in a petri dish by itself, and it divides every 10, 12 hours. So first there's 1, then there's 2, then there's 4, 8, 16, doubling. 1 week later, 30,000 cells in the petri dish. But the most important point is they're all genetically identical because they came from one parent cell. So I have 30,000 genetically identical cells, and I split them into 3 petri dishes, 10,000 in each dish. And this is going to come home real big in a second. In the lab, we grow the cells in a fluid called culture medium, okay? And let me just get a little hint here. Culture medium is a laboratory version of blood. If I'm growing human cells, I say, what's human blood made out of? And then I concoct a mixture of laboratory version of equivalent of blood. And if I grow mouse cells, what are mouse cells made out of? And the blood. And the blood is the culture medium. But since I make it in the lab, I can change some of the constituents. So I make 3 different versions of culture medium. Let's call them A, B, and C. Slight chemistry difference. I have 3 dishes of cells, genetically identical cells in all 3 dishes, but I feed them a different culture medium. So in dish 1, culture medium A, the cells form muscle. In dish 2, genetically identical cells, but in culture medium B, the cells form bone. And in dish 3, genetically identical cells, different culture medium, the cells form fat cells. And I'm looking at the results of these studies, recognizing I'm going to go in that classroom and teach everybody that genes control life. And here I am looking at this. I say, genes did not control this. Why? They all had the same genes. So I said, well, what made the difference between some cells going to muscle, some to bone, some to fat? And I go, it wasn't the genes. It was the culture medium chemistry. And all of a sudden it's like, well, wait. The environmental chemistry is controlling the genetics, not the genes controlling the cells. And then now we take one step back. And I go, as I mentioned, you have 50 trillion cells inside under your skin. You're a skin-covered petri dish. Inside your skin-covered petri dish, you have the original culture medium, blood. And I go, now here's the point. Does it make a difference if the cell is in the plastic dish or the cell is in the skin-covered dish? And I say, no. In this regard, the cell's fate is controlled by the culture medium, synthetic in the culture dish, original blood in your body. And I go, so the chemistry of the blood is controlling your genetics.
Thanks for watching. If you like what you just saw, why not subscribe? Here's a link to do that. And if you'd like to watch the full interview, you can find it at chrisbeatcancer.com. There's a link to it in the description right below this video.